Hey guys, welcome to the 2023 VCE Bio 3-4 lecture. My name is Angelica and I'm going to be your lecturer for today. So just a little bit about ATAR notes. We've been around since 2007, kind of as a way to even out the VC playing field and ensure that all students get a chance to fulfill their potential and do the best they can in their VCE studies. So we've got tons of free resources, which I'll probably discuss in a second, but if you haven't yet signed up for the other lectures, I would highly recommend that. They're really good, all run by students who've done really well in their studies and super informative and free, obviously, so I totally recommend signing up. So, just a bit about the ATAR Notes website. As I mentioned, we've been around since 2007, so we've come a long way. I actually personally used to attend these lectures in high school. Like, I literally remember going to this 3-4 bio lecture when I was in year 11, doing 3-4 bio, and they used to do them at RMIT Uni, and so I feel like I've come a bit of a full circle right now. So, yeah, really good place to get lots of free resources, really helpful. It totally helped me a lot in my VC studies, which I'll tell you about at the end. So, just a bit about our website. It's totally free. I recommend, if you're a bit self-conscious or shy like I was, making a fake name or a pseudonym or something and just registering. It's totally anonymous if you want it to be. No one needs to know it's you, and it's super helpful. So we've got lots of free study notes, which are downloadable and really easy to access. Everything's arranged really well and they're really organized. And so I really like them. They're obviously made by students who've also scored really well in their studies as well, which I really like. And because of this, I feel like they're targeted towards students as well. So I thought they were really good. We've got lectures like this one. You guys will be getting the slides at the end of it too. So if you're seeing another lecture that you want to attend as well, but maybe it's a time you can't attend or something like that, I recommend just putting your name down anyway and maybe having a watch of it later on or just flipping through the slides in your free time. So yeah, we have got a few others this week, so check out the timetable for that. And yeah, hopefully I'll see you on another one. So we've also got a Q&A discussion forum. This is what I found the most valuable part of the ATAR Notes website. So when I was in high school, I'm always super worried that this is not recording. It is recording. So when I was in high school, I didn't use the ATAR Notes forum for a really long time. Like, I remember going to these lectures and they were like, make an account. And I was like, okay. And I just didn't want to use it. I don't know. I felt really shy and I was like, I don't need help that badly, or, I don't need to put my questions on an anonymous forum or anything. But toward the end of bio, I started using the forums really hesitantly because I was kind of like, wow, everyone is so smart here. My question's going to get shut down or people are going to think I'm so dumb. But yeah, I put some questions on the forum and it can be a bit unnerving at first to do it, or a bit might feel a bit nervous about it. But everyone was really smart and helpful. They helped me out with my questions really quickly. And I remember, especially around exam time, people would just race to quickly answer questions. And I remember getting a response back in it was 27 seconds or something insane. And two people replied at the same time, and it was 27 and 34 seconds, and they were competing. But yeah, people are very on top of the forums. They really want to help you out with your studies, and a lot of it's really guided around helping you understand the question and do better in your own learning, as opposed to just giving you the answer. So I found it really useful to go through practice exams, and anytime I had a question that I was a bit unsure about or that I got wrong, I'd just pop it into the forum anonymously because I was too shy for anything more than that. And yeah, someone would get back to me really quickly, and it kind of helped me out a lot, and probably helped my teacher out too, so I wouldn't spam her with 50 million emails on a Thursday night. But yeah, really good place. I recommend it. If you take anything away from this lecture, it's either make an ATAR Notes forum account and use this discussion forum, or the purpose of mitochondria, which hopefully you guys know by now, is not the powerhouse of the cell. It's the site of aerobic cellular respiration. But yeah, in conclusion, sign up for the ATAR Notes accounts. It's free. No, oh my god, put in your email and your credit card and we'll pay later. None of that. It's literally free. So I recommend. Anyway, that's most of my spiel done. So we've got videos, which are really engaging, bite-sized little revision things, which I think are quite helpful too. Newsletters, which just help you stay in the loop, stay up to date with what's going on. We've got the ATAR calculator, which is a bit of fun. I would use all the uni calculators when I was in high school and just try and compare them. At the end of the day, though, you still have so much power to control your ATAR, because your exam hasn't yet happened, so yeah, don't waste too much time on this like I did. We've got lots of articles as well, so these are really cool because they always feel like journal entries from students sometimes, some student who scored really well might write an article about how they did really well in bio or PE or English or whatever, and they will define study tips or techniques and things which they use, which I find also quite helpful, and heaps more. So check out our website, you guys. Make a fake name and a fake account with fake email and everything if you need to, but I recommend it. Just check it out and have a look at it. Okay, on to our actual lecture. So we're doing Bio 3-4, so that's not what you're here for. This might be a bit overwhelming, but it might be overwhelming even if you are here for that too. So we'll try and go through as much as we can. I just want to preface this lecture with saying that I've deliberately put in like 100 slides in here because it's hard to find a good collection of study resources for the exam. And so I tried to put in as much as I can into the slide deck so that you guys have kind of a guide as to what to study for or how to get started in your study, or just kind of a really basic revision guide. So I probably won't get through as much of the content as I want to, and I might actually skip over some slides. I'm trying my best to get as much done, but I only have 2 hours, which is just not enough time for me to get through everything. So yeah, I hope you guys enjoy this. Let me just make sure I've got my timer on. But yeah, I've deliberately put in too many slides, so please don't say, oh my god, you've forgotten to do this many slides, or you haven't done enough, stuff like that, because I'm really trying to get through all of it, and it's really just to help you guys out, give you as much as I can. Let me just put my timer on. So, check how many minutes I'm at. Okay, 6 minutes. We're good. Yeah, on that note, I've also made sure to keep, try to keep some time spare at the end for study tips and techniques. So I might skip over some basic stuff like what's the function of a Golgi apparatus or something like that. But I'll try and keep more time for the study tips and techniques, which I think is probably more useful to you guys, because you guys have a textbook, you can read the textbook for definitions and stuff. But the study tips and how to prepare for the exam is probably something which I didn't learn until quite late in high school, and I find this to be the most valuable thing I took out of the lectures I attended. So yeah, if you've got questions about that, if you have questions about anything actually, just pop them in the chat throughout the lecture, and I'll talk about it. Well, I probably won't talk about it. I'll message you back in the chat with a response, and I'll try and talk about my personal study tips and things at the end of this lecture. So yeah, let's get started. Every time I see this, there's a black dot here and I don't know how to get rid of it. Annoying. Anyway, my name is Angelica, and I graduated in 2019 with a 97.25 ATAR. I achieved a 49 study score for bio, 40 for PE, and 47 for English. I graduated biomed, and I'm now doing medicine at Monash. So I did my 3-year biomed degree, and I'm doing postgrad med. So I'm really enjoying it. It's pretty full on, but I'm kind of a bio nerd, doing biomed degree and another medical degree now. And I'm I love it. It's so interesting, and I find it so fascinating. So if you've got questions about that, let me know in the chat, and I can reply to those as well. I tutor bio, PE in English, TootSmart, and ATAR notes, and I love my cats, and my dog. Long distance running. I love studying at cafes and reading there too. So yeah, let's get started. As I mentioned, any questions you have, put them in the chat, and I will reply to you as soon as I can. So we're covering a ton of stuff in the lecture, a lot stuff, so try and stick with me. I'm going to try and slow down my speaking. I've just tried to get through the intro a bit faster, but we have got a ton of slides to get through. So everything will be recorded and put down later, downloadable later. But yeah, I'm going to try and get through what I think is important or necessary or often misunderstood by students. So yeah, lots of things. Okay, firstly, this is a super common exam question. What is the function of DNA and RNA, and what are some similarities and differences? And you often get a question which is literally fill in a table discussing 3 similarities and 3 differences of DNA and RNA. So just knowing these similarities and differences is probably a good tip, because it's likely to come up in the exam. It usually does come up nearly every year. You get a question like, what are 2 similarities of DNA and RNA? Or fill in the table, or discuss how RNA and DNA are alike or dissimilar, or something like that. Anyway, some similarities we have include their phosphate backbone. They both base pair via complementary pairing. There are 2 bonds between A and T, or A and U, and 3 bonds between G and C. So we know that G and C are a stronger bond than A and T, because there are more hydrogen bonds between them. They're made of nucleotides, and they have an overall negative charge, which means that if we put them into a gel electrophoresis, they're going to gravitate towards the positive charge, or the positive electrode. Some differences include that DNA has a deoxyribose sugar, whereas RNA has a ribose sugar, which is in the name, deoxyribonucleic acid. versus ribonucleic acid. DNA is double-stranded, whereas RNA is single-stranded. DNA includes thymine, whereas RNA has uracil. DNA stays in the nucleus. So this is a really important one that we will explore soon. So it does stay localized within the nucleus, apart from some mitochondrial DNA, which we don't really talk about that much. Whereas RNA is often in the cytoplasm, and it actually has to leave the nucleus and go off to a ribosome. DNA is often really long as well, whereas RNA is quite a lot shorter and often more susceptible to degradation. I love these GIFs. They're so cute. Anyway, it took me a really long time to realize that ribosomal RNA is literally made of RNA. I know it's in the name, but it took me a long time to realize that the entire actual structure of a ribosome was actually made up of a long RNA, all intertwined and complex with proteins as well. So I don't know why it took me, I knew the idea, but it didn't click in my head until second year undergrad, and I was like, wait a minute, ribosomes are made of RNA. So yeah, this is showing ribosome RNA, this is inside of a ribosome. You'd have your ribosome made up of this. We also have tRNA, which is a type of RNA, oh yeah, there it is. So it brings the amino acids to the ribosome, transfer RNA, and then you've got mRNA, which is messenger RNA, which is read by the ribosome and really aids in the translation process. I know I've glossed through that, it's pretty basic, but slides are there, you guys have your textbooks and things, so yeah, maybe if you need more attention on that, spend some time on it. I'm going to go through some of this triplet stuff, codon stuff, and a bit about transcription and translation. So anything I've kind of bolded or highlighted, in a certain color, gene, or maybe not three, but DNA triplet, gene, RNA codon, these are all quite important terms, and I'd recommend actually making a glossary of them. And so I recommend making a flashcard out of them. I know Quizlet's paid now, but I think some Quizlet accounts can still make stuff out of on them. Otherwise, I know people don't like to use ChatGPT, but I think giving them definition and term and definition, popping a bunch of them in and say making flashcards, you can copy and paste them into a flashcards generator. So something like that could be really useful. Then you can print that out and use it, or even just making your own paper flashcards. I know you can get little decks of little cards, and you can get a ring to hold them all on, so you can flip through them really easily, which I used to do a lot in high school. So if you prefer that, I recommend it. Something to note about flashcards though is they're really great for memorization, really good for memorization, but it's really important that you understand the terms first. I'd often blindly remember flashcard words and not actually understand what they meant. You could tell me that it's a segment of DNA that talks with genes production or proteins production, and I would have no idea what that meant. So I encourage you guys to take the time to really understand each of these definitions. Break it down into English. So what is a gene? A gene's a piece of DNA that codes for protein, it's the instructions for making a protein. Break it down into basic English, pull it apart, and just have something that helps you understand it. When I make flashcards, personally, I often break these words down. If it's a weird Greek word polymer or something like that, I would have to put down the epidemiology, the etymology of it, and I might have to draw a picture to help me remember too. So kind of figure out the way you learn, and target that. So we'll get into study techniques later. I just maybe remember something I want to talk about there, so I might just write that down. Different learning styles. Okay, back to lecture. So how does DNA code for specific proteins? It's all contained in the bases. So 3 consecutive bases in DNA equals a triplet. So for example, if we had C, A, T, that's a triplet. When this triplet gets transcribed into an RNA codon, we've got G, T, A. So this would be the RNA codon. So the same kind of thing. So in the DNA, it's known as a triplet. In the RNA, it's known as a codon. Okay? Really important that you understand these definitions, because the examiners like to trip you up on this stuff. So each one of these RNA codons gets encoded into an amino acid. I'm not really sure what that encodes off the top of my head, but it's an amino acid. Let's pretend it's the only amino acid I can remember is citrulline. It's not a good one. I'm not sure. Anyway, histamine. Histamine? No. Anyway, we'll skip that. It's an amino acid, or it gets encoded into a stop codon. Okay, so make sure you understand your stop codons as well. If I don't have that written down, I'm going to go through a mnemonic for it. It's a bit stupid, but hopefully you remember it. Yeah. So this is the transcription process. I haven't written it here, but it's gene expression part 1, transcription. So transcription, okay, I'm not even trying there, but transcription is gene expression part 1. This occurs in the nucleus of the cell. Why does it occur in the nucleus? I know this is like pre-recorded, so you guys can't like shout out your answer, but it's because the DNA must stay sequestered in the nucleus. It can't leave the nucleus. And so the transcription process where the RNAs, or the messenger RNA is made, must occur in the nucleus. And that mRNA can leave the nucleus and go off to the ribosomes or be translated. So RNA polymerase, which is an enzyme, will bind to the promoter region of a gene to be transcribed into the template strand of DNA. Big words, they're all bolded, so I'd recommend you know what they mean. We'll try and go through some of them. So RNA polymerase, anything ending in A's is typically an enzyme. Polymer means a long chain. So this is an enzyme which makes a long chain of RNA. See how that just makes sense? Yeah, so it's better than just straight up memorizing what it means. Understanding it, pulling it apart helps. Binds to the promoter region. So promoter, if something's promoting something, it's allowing it to happen. So allowing this gene to be transcribed, allowing this gene to be read on the template strand of DNA. We'll look at this in a second. The RNA polymerase molecule actually contains a certain enzyme known as helicase, and it actually unwinds the DNA, moves along the template strand, and reads the template strand. This entire slide looks quite complex, but understanding this entire process and being able to like recite it, maybe not recite it, but explain the whole thing to someone else, even if it is like, I don't know, I have like stuffed dinosaurs that I will explain in my studies too. Like explaining it to a stuffed dinosaur or something, and trying to make sure that you understand it fully is a really good technique. So read through this all again later on after this lecture is done. Try and figure out if you understand the whole process, and try and describe the whole thing to your family or to your pets. I have to get my dog and sit him down and explain to him like, I don't know, metabolism or something like that. Or like a little stuffed toy, and just try and make sure you understand it. Anyway, so it reads the template strand in the 3' to 5' direction and makes RNA in the 5' to 3' direction, noting that strands are anti-parallel, which I'll show you guys in a second. The RNA polymerase will reach the end of the gene, which is known as a terminator. Terminate means to end, so that makes sense. You reach the end of the gene, you terminate it. And the pre-mRNA molecule will be released. And so this pre-mRNA molecule is complementary to the template strand and has the same sequence as the coding strand, except that T is replaced with U, because we know that RNA doesn't actually have thymine, it has uracil in place of it. So I just kind of explained this a bit. It's a bit wordy. Like, I need pictures to understand stuff. So we'll look at this here. So we can see here, this is the coding strand. This picture's a bit trippy because I usually think of it as the other way. But here, so we have the template strand. Remember that the RNA polymerase reads it in the 3' to 5' direction. What this means is that we've got the two strands of DNA. You've got your RNA polymerase enzyme with the helicase attached to it, which unwinds the DNA and opens it up so we can access it. What happens next is that, let me turn the little flashlight thing on. Laser pointer, torch. The RNA polymerase will then go and read this template strand in the 3' to 5' direction. Something else I want to note is that we've got the template strand and the coding strand. Okay, they might be switched in your exam. It might be coding strand underneath and template strand on top. Yeah, either way, reading the direction is really important. Knowing that the polymerase reads it 3' to 5', which means it goes from 3' to 5'. The reason why we've got 3' to 5' and 5' to 3' the other way is because it's anti-parallel. So this strand goes forwards in this way. This strand is complementary to this strand, but it's like flipped, So we've got A instead of T, T instead of A, G. Like, it's kind of like flipped, inverted in a way. So it's like the opposite is matching. So that's why you've got the sides. I think that was a really bad explanation. If you can just recall that we've got 3' to 5' and the other side must be opposite, 5' to 3', you're doing fine. That's all you need to know really for high school. Anyway, RNA polymerase reads the template strand. It uses the template strand as a template and it reads it in the 3' to 5' direction. So it goes left to right in this case. And it makes RNA, mRNA, in the 5' to 3' direction, which means that the 5' end comes out first. And what you'll notice is that this RNA polymerase molecule, when it makes the mRNA, it actually makes it really complementary to the template strand. And it actually makes it very, very similar to the coding strand. Only, instead of thymines, you've got uracils. So if you look at this, we've got, after reading this A, it's produced a U. After reading this A, it's produced a U. After reading this C, it's produced blah, blah, blah. And it just goes on. But if you look at the coding strand, you'll see that the coding strand is actually very similar to the new mRNA strand. And so we can see that it's pretty much the same, only that this T has been substituted for a U instead, okay? So you can see how similar they are. And so it's kind of a cheat or hack way to figure out what your template strand is going to give you for your mRNA. Like if I say, this section of mRNA right here, what is the mRNA going to read as? You just look at the coding strand and go, okay, well, it's going to be A, C, G, A, U, U. And so you've kind of figured it out really quickly. That's probably a good thing to know for the exam. Then it will reach a terminator region and just sees this transcription process. Complex diagram, but not really, because we've just gone through the whole thing. So have a go at reading this and working it out in your own time. Part 2 is translation. No, it's not. This is pre-translation. So I'm not going to name anything. Note here it says eukaryotic cells. Prokaryotic cells don't undergo any real types of post-transcriptional modifications like this within the nucleus. And the reason why is that prokaryotes don't have a nucleus, therefore they don't need to have these modifications added. So what happens in prokaryotes is that transcription and translation can pretty much occur at the same time. Whereas in eukaryotes, they're kind of sequestered in different areas. So translation occurs in the ribosomes, which are not in the nucleus, they're in the cytosol. Whereas the transcription must occur in the nucleus because that's where the DNA is stuck. Okay, so hopefully you guys understand that. The reason why we need these 3 types of post-transcriptional modifications is because RNA is often quite unstable. If you think about a virus infecting, you, right? This is not a good thing. The virus will inject its RNA or DNA into you, and this random free-floating nucleic acids inside of your body is problematic, and so it's often killed, right? Someone will go and chop it up, destroy it. And so your bodies have kind of been primed to think, oh wow, this random free-floating RNA is probably a bad thing, let's kill it. And so your body needs a way of distinguishing actual vital RNA and not killing it. And so we have these 3 modifications in order to ensure that we don't get this RNA to be destroyed. And so these modifications are introns are removed and exons are joined together. I think of it as introns are interrupting regions, exons are expressed regions. So we keep the exons and introns are interrupting, so we get rid of them. This means that the mature mRNA is shorter than the pre-mRNA. We add a methylguanosine cap to the 5' end of the RNA molecule. This is known as 5' capping. If you need to draw a picture, I would draw a little chain of RNA, and I draw it wearing a little baseball cap, and I have methylguanosine on it or something like that, and on a number 5 maybe, just so I can remember it. Maybe I'll do that real quick. I'm not wasting time. Number 5 wearing a baseball cap, which is MG, and this is RNA. I have to do these weird drawings to help me remember stuff because I think I'm a very visual learner. And so this is something that helps you out, I recommend. I know that looks like a B. Anyway. Pull the A tail is added to the 3' end. What this means is that we just have a long chain of adenosines, A, right at the end of the RNA molecule. Once these modifications have occurred, the RNA is known as mature mRNA, not pre-mRNA anymore, and it can leave the nucleus and move to a ribosome, which can either be free or attached to the rough endoplasmic reticulum. This 3rd stage here is going to be translation. I know I changed fonts halfway through. Translation. Okay, so in this stage we've got protein synthesis occurring, and this is the process by which a polypeptide molecule is produced from the mRNA code at the ribosome. What a complicated sentence. So, what is protein synthesis? It's where we make a polypeptide from the messenger RNA at the location which is the ribosome. So once that mature mRNA leaves the ribosome via a nuclear pore, It will then migrate to a ribosome, and the mRNA will enter the ribosome at the 5' end. Note the directions as well. It's really important for you guys to know that because it's often tested. So the start codon AUG instructs for translation to begin, directing for the amino acid methionine to start the polypeptide chain. Each successive codon in the mRNA will pair up the okay, it's getting a bit complicated here. Firstly, I want you guys to remember the start codon because this is something that you must know. It's in the study design. So the way I think of it is in American movies and books and things, they often start school in August. Now, we don't start school in August in Australia, so I'm not sure why I ever thought of this, but I was always like, oh, AUG, August. August is the start of school for American people. Let's go with that. So this could be completely false too. Maybe I'm reading these books wrong, but I just remember it as we start school in August, which is not true. But if it helps you remember, that's a good thing. So that's our start codon. AUG is our start codon. So if you see an AUG, it may be the start codon. Note that methionine is also often contained within the actual transcription sequence as well. So it could be just a random methionine inside the whole chain, but it may also be a start codon. The successive codons will pair up with an anticodon of a tRNA molecule. So a tRNA molecule is like a complementary little scanning region called the anticodon. It will scan the codon of the mRNA and go, wow, I match you. And it carries a specific amino acid to the ribosome. And so it like scans and goes, I match, let me give you my amino acid and we'll move on. So it brings that amino acid over. This results in lots of codons and anticodons pairing, resulting in the amino acid chain growing. And they grow and add to one another via peptide bonding. Okay, and I remember this because polypeptide is like the name for a protein, really. So polypeptides are formed via peptide bonding. Once a stop codon is reached, translation will cease and the polypeptide chain will be ejected from the ribosome. So we must know these stop codons, and I have a really weird mnemonic for this. So I tell this every year, and I get emails quite often from students telling me that this actually really helped them out. So if it helps you out, please send me an email or send me a message in the chat and let me know, because I like knowing that this helped people out because it's a pretty random mnemonic and it's a bit weird. But we have to know our stop codons. And so our stop codons are U, G, A, U, A, A, U, A, G. And so my mnemonic is like a visual scene, I guess, whereby a couple breaks up and they're fighting and they just like are really aggressive with each other and then they break up because they're saying some mean things to each other. And so my mnemonic includes the couple like yelling at each other. Like imagine some couple fighting with each other and they're going, you, go away. You are away all the time. You are gone. They're finally broken up. They're gone. Never see them again. So you, go away. Don't want to see you. U-G-A. I know it's like text speak, but U-G-A. You go away. You are away. Like one of them is always away. It's really ruining the relationship, right? U-A-A. You are away. And then finally, you are gone. Like the finally ended stuff. They were really toxic and not good, and it's probably for the best, but now one of them is gone. You are gone. And so these are our stop codons. So if you can remember this like vivid scene of you go away, you are away, you're gone, done. You've got your stop codons memorized, which is something which is really important for the exam. So well done. Hopefully this helps you guys out. I know it's pretty random and weird, but anyway. That's the whole translation process, which we've already done. Wow, time is going by so fast. Okay, I know you guys are going to be a bit mad here, but I'm not going to spend too much time on the trip Operon. But what I will do is direct you to some good articles about it. When I say articles, I'm not like meaning like fully intense scientific research articles. I mean the Khan Academy article, which has really nice cartoons. Khan Academy, if you look up Khan Academy trip Operon, it gives you a really good article. I'd literally like write this down in your notebook or in your notes app or something like right now because it's a really, really good article. It helped me a lot in school. I'd also look up the lac Operon, which is not on your study design anymore, but it does give you further understanding about different types of operons. And so VCA are a bit sneaky because it used to be that the lac Operon was on the study design. And then one year they asked about the trip Operon, which wasn't on the study design yet. And so I mean, I wouldn't put it past them to do that again. So making sure you have a sound understanding of operons in general is a really good thing. So we'll do a tiny bit of just revision on it. And I have got some like slides here which do go through a bit of detail. So that's something you guys can use for revision. But essentially an operon is a series of genes which are transcribed in conjunction with one another. So in eukaryotes, we can only transcribe one gene at a time. Transcribe that, done. Transcribe that, done. But in prokaryotes, you can transcribe a bunch of genes all at once. So you can transcribe all these genes all in one go. By the way, you guys, if you're writing it down, it's just Khan Academy article, trip Operon, and lac Operon. So lac Operon is for lactose. If you look up Operon, Khan Academy, it'll probably come up anyway. And it's just a really good article. Like, I remember specifically reading that article and going, wow, this explains it so well. Which would be like 5 years ago now. Oh my god, that was literally like half a decade ago. Okay, let's keep going. So we've got some methods of regulation in this operon. One of them is known as attenuation. So it represses, they completely prevent transcription from starting. So you repress this and, well, you have the repressor, sorry, I don't know why I'm pointing over here. The way I think of it is OR, or. So operator, repressor, so repressor will bind to an operator. And then you've got the promoter region there. So repressors prevent transcription from starting. So you can't have transcription actually starting if there is a repressor on the operator region. Whereas we've got something called the leader sequence, which is, here's the leader sequence, and you've got the attenuator encoded within the leader sequence. And so you'll have transcription occurring. When we transcribe this leader sequence, it actually formed mRNA, and mRNA is single-stranded. What this means is that inside the mRNA itself, you might have accidental complementary regions. Like if I have A, U, U, A, it can form like a little curl, right? A, U, U, A is not quite right, but let's say the sequence is G, G, A, T, A, T. We can actually have, like the mRNA is just, forming itself as like a piece of string, but then it goes, wait a minute, I've got like a complementary sequence within this. Let me form a hairpin loop. And so you've actually got the formation of a hairpin loop here at this attenuator sequence. And so what happens is that the mRNA, which is single-stranded, actually forms like a loop with itself, like a hairpin loop with itself. And so when we have the translations occurring, this can actually affect translation. Okay, I feel like that's the best way I've explained it in a really long time. So within this leader sequence, remember that leader is a bunch of mRNA molecules. They will be translated into codons, or into amino acids, right? But we've got, every group of 3 here is actually a codon. And so within the leader are 2 codons, which encode for tryptophan. So tryptophan is, we have to go on that, but it's amino acid. What happens is that in order to transcribe this leader sequence, this little loop, we actually need 2 tryptophan molecules. And so if we've got plenty of tryptophan molecules, when our ribosome translates it, it can kind of zoom through super fast because the tryptophans are there and ready. Whereas if we have a low amount of tryptophan, it means it takes a long time to actually go through. So it can be more careful in translating, and it's got to stop and wait for someone, the tRNA, to bring over the tryptophan amino acid, which is all fine. The problem with the fast one though, when it zooms through, because tryptophans are really readily available, is that the ribosome might actually fall off the sequence and it can disrupt translation. So in the case of low tryptophan levels, as I mentioned, the ribosome actually moves through that hairpin loop leader sequence very slowly because it's, waiting for the tryptophans to be brought over. So trip operon because it relies on tryptophan. This hairpin structure is created, does not stop transcription, so transcription can keep occurring. What happens there is when we have a high amount of tryptophan, the ribosome will zoom through, and what happens is that the hairpin structure, does stop transcription. It's just like, I think we actually have 2 different types of hairpin structures forming, so one of them is quite a slow, steady structure, whereas one of them is like a zoomy one. It's a bit dangerous to go through. But in essence, in the case of high tryptophan levels, the ribosome zooms through, falls off, and in falling off, I think of it as like causing an earthquake. And so we've got the RNA polymerase also falling off, and as a result, we stop transcribing. And something else to note here is that transcription and translation actually occur at the same time in prokaryotes, which I mentioned before, but this is where it's important. So because the ribosome's just fallen off so violently because it's been zooming through, because there's so much tryptophan, the RNA polymerase also gets shaken off. It's like, wow, earthquake, let me fall off. And so we actually seize not only translation, but also transcription of this operon. And the reason for this is because essentially this operon, the trip operon, makes tryptophan. If we don't need any more tryptophan often, like we can just shut down the system and just say stop, no more tryptophan. And the way it does this is because it zooms through. Okay, so hopefully that makes sense. I can't go through it again right now because we are a bit short on time. But if you have any questions, try and put them in the chat now, try and answer them. It's a pretty complex area of study. You don't have to go much more than this. Like this depth is pretty good. What we have in the slides is a really good explanation. Try and work through it slowly. Use the Khan Academy trip operon and lac operon articles, and you guys should be good to go. So hopefully that made sense, and good luck with this. I know it's a new part of study design too, so it's likely it will be tested. So make sure you feel familiar and okay with this. Okay, this is pretty basic stuff, so I might skim over it pretty quickly. But moving on to protein structure now. So we've got primary, secondary, tertiary, and quaternary protein structures. We've got a short description. Make sure you know that the primary structure is pretty much the sequence of amino acids in the protein. It's like a long chain of beads in a necklace. A secondary structure consists of alpha helices and beta pleated sheets. Something I want you guys to know is you must say beta pleated sheets, not just beta sheets. VCAR is actually very strict upon this, and if you just say beta sheets, you don't get marks. You should also note that we've got random coils. So we know that alpha helices look like this, sort of. Beta pleated sheets look like this. Like a corrugated iron type vibe. And we also have random coils. And so if this is our alpha helix, this is our beta pleated sheet, this little thing in between is a random coil. Tertiary structure is the 3D structure of the protein. It may be globular or, I can't remember the other word, like a linear type structure, fibrous or globular. And so it's kind of made up of the interaction between R groups of the amino acids. So what that means is when you actually have the 3D packing of the protein, you'll have like different bumps and ridges forming, and some regions want to be pulled to one another. And this is because you've got the R regions, or the variable regions of the amino acids, wanting to be drawn to one another. And this kind of helps create the actual 3D structure with a clumping. So for instance, if you have a sulfide ion here and a sulfide ion here, when you join, when you're making the whole thing and it's folding up into a 3D structure, those two sulfides might want to be close to each other to form a disulfide bridge. And it kind of helps form that 3D shape. Quaternary structure is the 3D structure, but if it's actually within another 3D structure. So if you have several polypeptides put together, this creates a quaternary structure. Make sure you know the types of bonding and folding occurring at each stage. And yeah, this is a good table, you guys. So yeah. Okay, moving on to enzymes. So enzymes are what? They're a biological catalyst, which is something you should know. So remember this definition, biological catalyst, which means that they speed up the reaction rate of reactions, of natural organic reactions typically. Anyway, so enzymes, biological catalysts which speed up reaction rates. Here's an enzyme, here's a substrate. So a substrate will kind of bind into its little pocket here, which is known as the active site, and to form an enzyme substrate complex. And then this enzyme substrate complex involves the enzyme kind of enacting some kind of transformation, I guess, onto the substrate, and this results in the production of products. You might actually have the process looking a bit different. You might have two separate components entering that substrate complex and then being expelled as a singular product. Either way, the enzyme facilitates this conversion. A type of particular enzyme is an endonuclease. Note that it ends in ase. Endonucleases are also known as molecular scissors, colloquially. So if you're going to say this in the exam, you'd probably have to put it into quotation marks, but I wouldn't recommend you use this as your definition. They're also known as restriction enzymes. And so they're pretty much molecular scissors which cut DNA at specific base sequences. And so remember we've got our DNA backbone, which is made of that phosphate backbone, or the phosphodiester linkages. We actually snap that. I don't know, I can't just snap my fingers. Anyway, we snap the bonds there and it breaks. So if we have say the DNA is continuing on up there, we actually snap the bond there and break them into two separate pieces. So base sequences which cut are palindromic, which means that they're the same forwards and backwards, like the word race car. And so we can either get sticky ends or blunt ends. And so blunt end is where it's just straight up and down. Whereas a sticky end involves this little overhang. And the reason that sticky ends are better than blunt ends is because they actually facilitate stronger bonding. So for instance, this bit here, it has no specificity, it has no overhang. So this can like bind over here, it can bind over here, it can bind over here, it can bind anywhere, right? It has no specificity. So if you have one, chain of DNA, which has just been snapped open, another chain of DNA, and you want these two to join together, you might accidentally have this bit joining here and this bit joining here because there's no like loyalty to binding to its own DNA in this case. Whereas with the sticky end, you actually have the overhang, which means that, because you've got a C here, you must have a G complementary to it. You must have a T being complementary to an A, and then etc. Like, it must be complementary. So it kind of facilitates that they will only bind to what is complementary. And so you can be sure you won't accidentally bind to another random string of DNA. Cool. Ligase, molecular glue. So it joins those two strands together. So I know we have the complementary stuff, which makes it better binding. We also add the ligase just to seal it in properly. So it can form one longer strand of DNA. And it catalyzes the formation of that phosphodiester linkages, which is a covalent bond. You don't have to spend too much time knowing this in bio, but just understanding that a covalent bond is where two molecules share electrons. So it's quite a strong bond. Cool. Polymerases. So, polymerases are enzymes that join nucleotides together, blah, blah, blah. We know, if we break down this word, polymer means a long chain. Ase means enzyme. In essence, this is an enzyme which makes a long chain of a particular molecule. If we tack the word DNA on the front, then it's an enzyme, which makes a long chain of DNA. Or if it's RNA polymerase, it's an enzyme which makes a long chain of RNA. Okay? So, try and break down the words. I find that's really helped me in biology. Okay, now we're going to go into CRISPR-Cas9. And so this is a really interesting and revolutionary aspect of biology and like STEM technology, which I think is really fascinating. STEM technology, it's kind of an oxymoron. Not an oxymoron, what is it? Just a waste of words. Tautology. Anyway, it's technology in science that we use and is really radical and is transforming the STEM world, really. So first we need a bit of background. So this is a bacteriophage. And so a bacteriophage is a type of virus that infects only bacteria. The bacteriophage lands on you, cool. You're not a bacteria, so you're not the target. But if you're a bacteria, like Pseudomonas aeruginosa is, which is implicated a lot in burns, it can actually infect that bacteria and like kill it. So what happens is that this virus or bacteriophage lands in the bacteria. I know I'm going really fast, guys. I might give you like a second just to like breathe, like get a drink. Literally like 10 seconds, I'll get a drink too. But hopefully you guys can like rewatch this and like slow me down where you need to. Cool, okay. The time is going by so fast. Crazy. Okay, let's get started again. So bacteriophage injects its viral load into the bacteria. And this is going to be either DNA or RNA, because we know that viruses can give either DNA or RNA, and this likes to insert itself into the genome of the organism it's infecting. So, in this case, in this case we can see that this, I don't know what that is, it could be DNA or RNA, it's probably RNA if it's single-stranded. Anyway, this virus, bacteriophage, has infected the bacteria, it's put in its genetic material, and it's trying to make the bacteria sick. And so bacteria don't have evolved immune systems like us. Like, we have a very fancy immune system. Bacteria aren't as sophisticated because they're prokaryotes. And so prokaryotes have quite a rudimentary immune system. But we've actually stolen this immune system and we're using it to revolutionize the STEM world. So we'll look into that. So what happens is that the bacteria gets sick, it's really, troubling and sad, but what happens is the bacteria is smart. So Pseudomonas aeruginosa or whatever other bacteria goes, yeah, I got sick once, I'm not going to get sick again. So I'm actually going to take a copy of this viral load, this RNA or DNA, and I'm actually going to keep a copy of it in my little library of DNA or RNA. And so what happens is that they actually have an enzyme known as CRISPR-Cas complex. So I'm pretty sure the Cas is the enzyme, the caspase, just like the molecular scissors, it's an endonuclease. And CRISPR is the CRISPR array, which kind of stores this, I think it sort of stores that copy And so it like reads the library of genetic material here. So what happens is that this bacteria gets sick, it's sad, it's hurt, it's sick, and then it recovers, so it's fine, goes to the manors. And then what happens is that bacteriophage actually lands again and injects its viral load in, and this bacteria is like, no, you've infected me before, I know you. And so this CRISPR-Cas complex actually goes and checks the library and goes, yes, we have a copy of this virus's genetic material from before, let's take a copy of that, and they kind of like hold on to it, and they make sure it's complementary. They actually make a complementary copy to that viral load. And so what happens is that they can actually scan any invading RNA or DNA and go, wait a minute, I've got a copy of you from my CRISPR array, this like library of collected DNA and RNA. And because I'm complementary to you, I know you must be a foreign, evil piece of material here, so I'm going to kill you. And so the caspase actually cuts up that viral load and renders it ineffective. And so it actually protects the bacteria from like subsequent infections. So the first time it gets sick, it takes a copy of that, viral genetic material, and the next time it remembers. So it doesn't have like an immunological memory, the same as we do with our antibodies and things, and like plasma cells, but it does have this very rudimentary system, which relies on finding complementary pieces of genetic material and cutting them up. And so we've actually stolen this for use in cloning, or just genetic modification technologies. So if we got like a tomato plant and we wanted to make it glow in the dark, we can actually insert a protein from a jellyfish. I can't remember the name of it. GLP-1, I think. Anyway, this GLP, I'm pretty sure GLP. You can insert this gene into a tomato plant. And so what happens is that you can get a CRISPR-Cas9 complex, make sure it's complementary to a particular region of the tomato plant, and cut it up. And then you can insert, make sure it has sticky ends, insert that glow-in-the-dark green gene, make sure it's also cut the same sticky ends, and then ligate the whole thing together. And you actually have the insertion of this glow-in-the-dark green into the tomato plant, right? And so we've actually stolen like this CRISPR technology from bacteria, from their like rudimentary, not as sophisticated as ours, immune system. So it's actually a very cool system. That was quite a short like tangent on that, but hopefully that kind of puts into context. The best way to study for this area of study is to look up like articles. Like I personally really enjoy the news app on my phone. Like I have an iPhone and just the news app, because you can actually go to like topics and search up what topics you like. And I really enjoy biology, so I've obviously favorited the biology topic. But they often have articles about genome technologies or particular medical revolutions. Things like Ozempic, you might have heard, is a drug which is for diabetics initially, but it helps with weight loss. And so it kind of simplifies this kind of complex, often jargon-heavy articles and things into kind of general speaks that the general audience, general population can actually understand it. And so they allow you to get a broader understanding of these different topics and areas or issues. And so like looking up CRISPR-Cas9 technology or CRISPR, or literally just looking up CRISPR will bring you a bunch of results. And so just reading these articles or think pieces debating the pros and cons of CRISPR is a really good way to study for the exam, because it helps you to kind of contextualize how CRISPR can fit in the wider evolution of genome technologies and the human race as well. I know I'm kind of going on a bit. You might be like, why is she telling us this? In conclusion, your exam often requires you to discuss the pros and cons of CRISPR and other genome technologies. So reading other articles about it and getting a broader understanding of why it's good, why it's bad, why is it ethical or unethical, help with your basis for your argument. And so VCA have been gravitating towards more essay-based questions or longer extended response questions for these topics in recent years. So having a greater understanding of it will help you with your exam. Okay, lots of talking. Polymerase chain reaction is a technique used to amplify segments of DNA. We need Taq polymerase, which is taken from Thermococcus bacteria, which live in hot springs, DNA primers, which kickstart the replication process, and nucleotides. So we start off with a double-stranded piece of DNA. We might be at a crime scene, and we might find a tiny bit of DNA, and we go, this is the criminal, but we don't have enough DNA to work with. So what we do is we amplify it. So we get that DNA, extract it, and we actually heat it up to 94 to 95 degrees. I say 95. For the annealing stage, you can use a range. For the extending stage, make sure you use 72. Anyway, stage 1, heat it up to 95 degrees. This is near boiling point, and so it actually separates the DNA into two different strands. Next up, you've got the annealing stage. You add the primer, So primers are like little complementary regions which start and like bind to the DNA and kickstart the activation process. And so you've got the primer, you add the Taq polymerase, which can withstand high temperatures, which is why it's used. And then you also add your free nucleotides. So you add all these things in at the annealing stage, and at the extending stage, you heat it back up to 72 degrees. Actually, you might add these things later on in the stage after the primers have annealed. So you add your polymerase here, you add your free nucleotides, and you heat it back up to 72 degrees, which is when Taq polymerase is activated. And then it starts the extension process. And what happens is that you end up with 2 copies of DNA. So you start with 1, end up in 2. And so this is, oh, what's the word? I had it in my head a second ago. It's like a methods word where you double. Exponential. It's exponential like reproduction, I guess, of this DNA. So it kind of doubles every time. Okay? So you end up with 2 strands at the end. When you do this whole process again, which is quite quick, you end up with 4 strands, or 4 copies. Double it again, 8, 16, blah, blah, blah. And it keeps getting bigger and bigger until eventually you've got like a whole mass of DNA to work with. So lots to work with, really useful. PCR is amazing. We also know that we use PCR in COVID testing. And for a long time, I didn't understand why, like how are we doing this? What happens is that you go get your, PCR test at the car park or at the hospital or wherever you do it. And they swab your nose, get your DNA. And what they do is they put this DNA into a test tube, heat it up, add some primers. And these primers are specific for SARS-CoV-2, whatever variant is out and about, right? And so they add some primer. If the primer doesn't bind, it means it's not COVID. Like if there's nowhere to bind, there's no COVID present in this entire segment of DNA which has been supplied. If the primer does bind, then you've got the whole replication process correct. So if you have many copies of DNA being made, you go, many copies are made. This means that the primers must have bound because we can't have extension without primers binding. As a result, if you have got this production of many copies of DNA, you know, okay, this person has this particular variant of COVID or has COVID. So you can say yes. Whereas if the primers don't bind and you have no new DNA being made or no copies of DNA being made, you can go, okay, well, no, they probably don't have COVID because the primers did not bind. We do not have this extension phase occurring. And therefore no replication of DNA. So hopefully that kind of puts into context PCR and how it works. Next up, we've got gel electrophoresis, which is a technique used to separate DNA fragments by size or weight. So we've got 2 electrodes, negative and positive. Make sure that you understand that DNA moves toward the positive electrode because it's negatively charged and opposites attract. It's often asked in the exam. So what we do is we add the DNA into little wells. We often add a color to it. So a common exam question is, nothing was observed. It could be because the DNA is essentially like transparent or just invisible and you can't see it. So you have to add either a stain or like ink to it so you can see it. Sometimes a stain is only visible under UV light, so you might need to expose it to UV light. Anyway, so you add the DNA plus stain into the wells, you turn on the charge. Within this agar rose gel, we actually have little pores, and the larger pieces of DNA take a long time to slip through. Whereas the ones which are small can obviously skim through those little pores and get to the end a lot faster than the other ones. Okay? So make sure you stain the fragments. We can also use like PCR and gel electrophoresis in DNA profiling. So I kind of mentioned criminals. You can also use it for like paternity testing and stuff as well. So we have these things in our genome called STRs. And what they are is that at particular locations of our genome, we have just random repeats. So you might have like DNA, like for instance, I might have the DNA sequence cat repeated 5 times. Whereas you might have it repeated 7 times. This literally like means nothing. To like no difference, no changes in anything really as far as we know. It's just everyone has a random number. But what happens is that you actually get this random number from your parents. So you've got 2 copies of this. One from your mother, one from your father. So if I have cat 5 and cat 6 copies, like 5 copies and 6 copies, then my mother maybe had 5 copies and my dad had 6 copies. Okay, whereas you might have 7 and 3. Okay, one from your mum, one from dad. Anyway, the point is we all have these STRs. There are quite a few of them inside of our genome which we can use, and we can actually use it for DNA profiling. And so if we find a DNA sample and we look at it and we actually look at this particular locus, this STR locus, we can go, okay, there are 3 copies and 7 copies of this STR. let's compare it to, like, say you're a criminal. No, say you're a child who doesn't know who their father is. You can actually compare it to possible people who you think the father might be. And if you look at it here, this is an example. So, say we know the mother here. Yep, this is the mother and this is the father, or someone we think the father might be. So we know this is the mother. We're not sure about the father, but here's a potential suspect of who the father is. And so we look at this child's STRs, and so we can go, okay, this one STR. So I think I said we've got two. We have one. No, as far as you guys need to know, we just have one. Like, I don't think we need to get into the complex genomics of it. You might get to uni and learn something different. But anyway, we have one copy from either from our parent, like our mother or our father, okay? So, one copy of this STR from our mother or from our father. This one, for example, you can see it matches with the mother's one. This one, for example, doesn't match with the mother. So what we would do is we'd look at this STR and compare it with all possible, like, dads, right? And go, okay, if this matches with this man, he could be the parent. If this one doesn't match with, like, this man over here, he cannot be the parent because it must have matched with him. This one's the mother, so this one is not the mother, so it must be with a potential father. And so you don't have to have all the father's ones matching. You need to have all the child's ones matching both parents, okay? I think the FBI uses like 17 different loci STRs. So what this means is that most people have completely like different numbers. Like I would likely have different numbers of this from my siblings, even though we're both related. Because I might have like 5 from one locus from my mother and 7 from another locus from my father. Whereas my siblings might have different numbers at each locus. So unless you're a genetically identical twin, you're not going to have like the same STRs at each locus. But people incidentally do, and so raising the number of loci that we look at can actually increase the validity of results. Instead of say like, because I think in a court of law, you can't actually rule out if someone's like guilty or not. I think because there is still the possibility that someone else, like if you find DNA which matches someone, you go, this person's guilty. You can't really say that just by looking at the loci, because like you could have someone else who looks exactly the same and just so happens to have the same loci and same STR repeats at each loci, and so it's kind of like such a tiny, tiny, tiny 1 in billions possibility, but you can't rule that out. Anyway, I think I spent too much time on this. Hopefully you understand this process and we can move on. Okay, GMOs. Genetically modified organisms that have had their genome artificially altered. It's a very bad box. GMO. Transgenic organisms are GMOs which have had a gene from another species inserted. So once again, if we've got a tomato plant and I put a jellyfish gene in this tomato plant in order to make it glow in the dark, this will be a TGO. It will also be a GMO. Okay, if I get a fish and I just cut out one gene for no reason other than I don't like this gene or the gene makes them sick, I have a genetically modified organism. But because I haven't inserted another gene from another species, it's not a TGO. That's all you really need to know here. But make sure you understand it because it does come up in the exam. Okay, we talked about enzymes previously. We talked about that. We haven't talked about denaturation yet. But denaturation is a process whereby hydrogen bonds are broken. And if an enzyme is exposed to high temperatures or pH levels that are outside the optimal range, it can actually denature. And what this means is that the protein kind of breaks down and the active site's kind of ruined. And as a result, it can't facilitate its general reaction, which is, converting the substrate into a product. So high temperatures and pH changes can break these hydrogen and ionic bonds, and denaturation is permanent. And so the example they often say is you can't unfry an egg. So eggs are full of protein, enzymes are proteins. If we fry this egg by exposing it to high temperatures, we can't like unscramble it, It's kind of destroyed. So it can't catalyze its reaction anymore. Okay, I noticed this before and for some reason it hasn't fixed. Oh my god, we're at 35 of 115 slides. We're going to try to get through a bit faster, guys. So we've got 2 types of enzyme inhibition. Competitive inhibition. So competitive is where you've got an inhibitor which has a similar shape to the actual substrate, and so it competes with the substrate for the binding site. Once it binds, it blocks the substrate from binding. The next one is non-competitive inhibition, whereby that inhibitor actually binds at a region known as the allosteric site, which is elsewhere. So it binds to this allosteric site here. As a result, it actually causes a conformational change in the shape of the active site, which you can see here. And so, as a result, substrate cannot bind even though it's free to bind, this is now no longer conforming to the substrate shape, and so it cannot bind there. Okay, so it kind of disturbs the binding. Here we've got different factors which can affect enzymes. We can see that different enzymes in different regions of the body are affected by different pHs. So in our mouth, we've got a pH of about 7, which is where amylase, the enzyme which breaks down carbohydrates, works, whereas in the stomach, we've got a 2 to 4 pH level. And so pepsin works there. So if I put pepsin in your mouth, it won't work. If I put amylase in your stomach, it won't work. In terms of temperature, just noting here, anything too high or too low above the optimal pH can denature it. In terms of temperature though, if the temperature is low, your enzyme isn't denatured, it just works slowly. Once you get to a high temperature, it will, like the optimal temperature, it will work really well, but after this, it will denature permanently and it won't work anymore. Okay, moving on to photosynthesis and respiration. So we've got different coenzymes, known as ATP, NADH, and NADPH. Something I want you guys to remember is that the NADPH is the electron carrier molecule in plants. I just think P for plants. And so what it does is that it carries hydrogen ions and electrons and facilitates either light-independent reaction or the electron transport chain here. And ATP pretty much gives us the energy for like everything we do. So it's the energy currency of the cell. So the light-dependent reaction, this occurs in the grana. And so grana is, I have a picture, I don't have a picture. Make sure you know where the grana is. I'm pretty sure it's a stack of thylakoids inside of the plant cell. And so what happens is that light energy is absorbed here. You actually have oxygen molecules forming O2, and hydrogen ions and electrons are collected by NADP. I think of these as baskets. NADP is unloaded, NADPH is loaded. So you've got a basket which is empty here, and then once you fill it with hydrogen ions and electrons, it becomes NADPH. Maybe think of the curve. Yeah, I don't even know. Just think it's a basket. Now it's a loaded basket full of electrons and hydrogen ions. There, energy harnessed by the chlorophyll is used to form ATP. So we actually form some ATP. So in conclusion, during the light-dependent stage, we have ATP and NADPH forming, which is then used in the light-independent reaction, which occurs in the stroma, which is the fluid-filled space surrounding the cell. So stroma is commonly referred to as the Calvin cycle. In a series of reactions similar but opposite the Krebs cycle, carbon dioxide is turned into glucose. And so the ATP produced in the light-independent state provides the energy-drive reaction. And you've got rubisco, which is a really important enzyme. Okay, I'm just going to open a really good article. I just looked up rubisco enzyme one time, and I found this really good website. This one, PDB 101, has really good explanations of different enzymes. It's kind of a lot of reading. You don't have to read down there, but just reading this general thing is a bit overwhelming, but it actually gives you a really good background as to what rubisco is. And I think I've used this website for other things as well in undergrad. So there are lots of different molecules. So that's a molecule come across if you do biomed. But yeah, I think it's a really good website. I like the little pictures and I like their explanations. So have a look at it for rubisco. You can rewind this thing. Otherwise, you can just look up rubisco enzyme and it will come up. Got less than an hour left. But yeah, good website. Understand rubisco, understand why it's important, because this is new to the study design, so once again it's likely to be asked by the examiners. I'm not going to spend too much time on this because we don't have a lot of time, but know about C3 plants, which are our general plants, C4 plants, which separate the light-independent and dependent stages into different cells. I know I'm just talking, but I'm going to give you a list of things to know for each stage. So for this stage, knowing what type of plants, and the process, how do they do this? So try and do like a flow diagram or something to draw this out is a good way of understanding it. In the case of CAM plants, these separate the light-dependent and independent reactions over time, meaning that like during the night you have the stroma being open, stroma? I can't remember the name of these holes. Anyway, these things are open. The stomata are open at night. This allows carbon dioxide to enter, so you have the first stage. Actually, you don't have the first stage occurring. You've got the first stage occurring here where you've got light needed, and then you've got independent light reaction happening here. And so just understanding this process too, and understanding what types of plants are using this process is also really important. So I know we didn't do much photosynthesis, guys, and I'm sorry. But hopefully you guys have covered that a lot. Make sure you understand that general stuff I just listed. Go and look at the Rubisco enzyme and how it works. In conclusion, it's not a great enzyme. But because I have so much of it, because we've got so many plants on Earth, and all these plants have so many Rubisco, it kind of works to an adequate degree. Okay, moving into respiration. So this is cellular respiration now. The function of respiration is to break down glucose into a usable form of energy for the cell. So we've got this big diagram. I'd actually recommend, I think the ATP concentrations may be off. So I would actually double check this. That's probably not off. But the overall number, it looks like the overall number you can't see anyway. Can you see it anyway? Well, this 26 or 28 might be different. So I would actually, you guys, like, go and double check this with VCA and make your own table. So make your own table. Like, this is a good guide, but double check these numbers, because if you look up the VCA, oops, Command T. Oh no. VCE Bio 3 and 4, Q and A, Q and A. This is a really good document. If I can find it. Oh, it's on the VCA website. So this is a study design, but there's a Q&A document. Yeah, and I would recommend going and having a look at that. Here, FAQ, frequently asked questions, sorry, not Q&A. So this support material, go and have a look at this, because it actually gives you a very clearly defined number of ATP produced for each stage. I might just quickly open it now. Just like super conscious of time, so we might try and skim through the rest of the content quite quickly. This is also a really invaluable document, so I know people might tell you to do stuff and it's like, yes, okay, sure, but this is a really good document. I recommend reading the whole thing. Look, it literally defines all the trip operon stuff you need to know as well. So, like, read it, it's really good. And then in terms of, here it is, it gives you the amount of ATP produced. Okay, so I think my diagram might be right in that 26 or 28 ATP produced here from the electron transport chain. Okay, we might be fine. But double check that anyways, so just be careful. I've also noticed when I was in high school reading textbooks and things, and sometimes they'd have mistakes as well. So just read everything, double check with the VCA study design as well, because a lot of them just detail what, what might be true, but VCA might use this particular number or round it up or down. Okay, we are going to go through this super briefly. So if you were here for the photosynthesis and respiration part, I'm sorry, we're not spending too much time on it, but what I would recommend is go back to our previous lecture series. So I'm pretty sure I did cover, so I'm not sure if I did 3 for bio, but if I did, if someone else did it, they're going to cover photosynthesis and respiration in a lot more detail than this. So go back and have a look at it. Yeah, so glycolysis is the stage where we break down glucose. It's the first stage of both the aerobic and anaerobic pathway of respiration. So we break down glucose. Second stage is the Krebs cycle, where we produce coenzymes, which go into the electron transport chain. So remember, we produce coenzymes in photosynthesis. So instead of producing NADPH here, we actually have the loaded NADH form. NAD becomes the loaded basket NADH, which carries lots of hydrogen ions and electrons. So I'll spend a bit of time on this slide because this one is a bit better. So in the electron transport chain, or it's more important, I guess, we have electron carrier molecules giving up hydrogen ions and electrons at the cristae of the mitochondria. The electrons are accepted by and passed along a series of cytochromes on the cristae, and the interaction between the electrons and protein complexes facilitates the production of ATP. So oxygen captures. I don't like reading stuff, like I don't think it absorbs my head. And I was going to tell you guys at the end in study tips, but I think just spreading it throughout is probably better. I personally prefer, like, I can't listen to something and remember it. Like, I even find it hard to memorize songs in the car. Like, I know some people are really good at that. And if you're good at that, I recommend listening to podcasts and stuff. But, like, I would try and listen to podcasts on my way to uni, and it doesn't stick in my head. What I need to do is draw stuff out or read it from a textbook, which might sound really boring, but I need pictures. So I'm going to explain this. So we have a series of cytochromes, which are pretty much protein complexes embedded in the matrix of the mitochondria. This is where the electron transport chain happens, okay, in the cristae, which is the folds. So it's not in the matrix. The matrix is in here. The cristae has these folds, which have these, proteins which span the entire membrane. What happened is that we had NADH produced in the Krebs cycle. Lots of loaded baskets. And essentially, we just throw these loaded baskets at these protein complexes. And we just pass a bunch of hydrogen ions along these protein complexes. So we pass the hydrogen ions and electrons along this protein complexes. So we pass a bunch of electrons. And so it pretty much makes a chain of electrons being passed from one cytochrome protein to another. And they keep being passed along. And what this causes is it kind of generates a gradient of hydrogen ions, and it actually turns this rotor on this enzyme known as ATP synthase. ATP synthase is kind of an insane enzyme because it literally makes ATP, which you might be like, okay, cool. But this literally exists inside of us, like at a microscopic level inside of our mitochondria. So I'm going to show you a really cool diagram, which I just think is like amazing. Every time I like tell someone about this, I have to be like, look at this. Isn't this the coolest thing you've ever seen? So this thing is inside of us, and it is spinning around and literally turning like a mill or something and making energy like right now, like constantly for your entire life. Okay, that's not as cool. But this one, watch how it spins. It's so cool. That's like there were mitochondria all over your body, right? Making energy for you. So just imagine all the muscles in your body. If you do lots of aerobic training, right? Like if you run marathons and stuff, you actually get more mitochondria. So you're making more of these proteins, or these enzymes, which turn and create ATP energy. Anyway, I hope you guys are as like impressed by that as I am, because I think it's very cool. Hopefully that makes sense. Something else to note here is we have water being produced. I think I see water on this diagram. But anyway, you need to know that water is produced here. Because often you see there it is, in the respiration formula that water is produced, and it's like where? It's the final stage. ETC, electron transport chain, is where water is produced. This is something Vika likes to ask randomly. I feel like it's a low yield question, but anyway. So I mentioned CRISPR before, so this is just a bit more about CRISPR. It can be used to edit genomes of certain plants. So I mentioned you can use it to delete genes, and you might actually have genes which promote disease. And so some plants or animals or whatever might have genes which promote disease, and if you delete that, then you're less likely to get the disease. Or you can like edit a gene and make it more like fruitful, right? You might have a plant which you can edit and it makes it like, fruit more. Maybe really good. Okay, I have a really brief thing on biofuels. I know it's literally one slide with 3 sentences, which isn't great. The thing about biofuels is that they're a new and kind of evolving field. This is another area of study that Vika likes to ask extended response questions on. So once again, I'd actually recommend you guys look up, why are biofuels good? Why are biofuels bad? Why are biofuels unethical? Why are they better for the environment? All of this stuff will help aid your extended response, okay? Because it's a newer area of study. As I mentioned before, Vika likes to move toward extended response essay almost questions. They're not real essays, but they're like pretty much a paragraph defending your argument. So making sure you understand this area is really important. And I'm sorry, I don't have much to give you here. But yeah, spending some time reading news articles, looking up like things like Wired magazine. I really love The Atlantic. It's one of my favorite magazines. They like to discuss stuff like this. So yeah, just look up Biofuels Explained, or The Conversation, which is a really good Australian run like a news network, but it's all kind of sciencey topics, which is really cool. Okay, on to our favorite topic, immunity. I actually love immunity. I think it's so cool. Every year I do immunity and I'm just blown away by how much more difficult it gets. I'm just like, what? Who knew immunity was so difficult? But yeah, I think it's a really fascinating area to study. So these are just different pathogen types. Obviously know your bacteria and viruses and how they work. This one that says protozoa should actually be prions. So I know it says protozoa here. That should say prions because protozoa is here. So prions are pretty much a single misfolded protein which can actually just wreak havoc inside of your brain. So you can get prion diseases from, well, I think actually Alzheimer's and Parkinson's are considered prion diseases. And you actually have the build-up of these plaques beta amyloid plaques and tau proteins inside of your brain, and it causes the loss of like neural tissue, and as a result you like lose your memory and things like that, which is quite awful. You can also get prion diseases from eating another person, which is another reason why cannibalism is not a good idea. But also mad cow disease, so eating infected like meat which has a prion in it, you can't, like if you do that you will die. So a few years ago there was a big mad cow disease outbreak and they had to kill a bunch of cows, which would have been like eaten for meat, but we couldn't eat the meat because they were infected. Finally, on eukaryotic pathogens, you don't need to know too much, just know that fungi has like a really thick cell wall made of chitin, or chitin, I'm not sure to say that. Protozoa are like parasites like malaria protozoa, like a little pathogen which gives you diseases. Worms, such as tapeworms and fluke worms, sugar IgE antibodies, I think. And yeah, and these are harder to treat than prokaryotic because they're eukaryotic, which means they're much more similar to us than all these other things, which are either prokaryotic or just not even alive, really, like these two. Okay, innate immunity is just like the immunity we are born with. If someone's innately good at the piano, they were just born with this ability to play the piano really well. Whereas adaptive is something that grows over time. So innate immunity is like, we have skin. We are born with skin. Intact skin is a physical barrier against pathogens. Okay? Adaptive immunity is like, okay, I was exposed to SARS-CoV-2 last winter. As a result, I've built up immunity against SARS-CoV-2. My body remembers it. Next time it hits me, I'm going to make antibodies against it. And hopefully my response will be a lot faster and less severe and intense. So adaptive immunity creates memory, whereas innate is like no memory. Your skin doesn't go, oh, I've seen this disease before. It just blocks anything which looks suspicious anyway, because it's a physical barrier. It just blocks stuff. Okay, I don't know the pictures over there, but know your first line of defense. This includes physical, chemical, and microbiota barriers. So we've got earwax inside of our ears, which literally clog up the ears and prevent stuff getting into your actual brain, I guess, or your nose or any other regions. You've got tears, which have lysozymes in them, Lysozymes, lysozymes, which are different to lysosomes. You've got saliva, which has like some pH in it. Sneezing, intact skin. You must say intact skin, because students lose points on this every year. Intact skin, because if it's just skin, you might have a breach in your skin, you might have a cut, which a bacteria can get into. Microbiota intestines, urine flow and vaginal pH. Different pH levels can destroy things which aren't meant to be there. Sneezing, we've got this thing called the mucociliary elevator, which, you know, when you get a tickle in your throat and you cough, maybe some pathogens tried to get in some dust, and your body's like, that's not meant to be here, so it coughs it out. And you might actually like cough something out with it, which is mucus, because it like sweeps it out and clumps it through the mucus and throws it out. Etc. Know that first line of defense is different from like physical barriers. Like stomach acid is not a physical barrier, it's a chemical barrier. So make sure you understand that. Okay, distinguishing between self and non-self. So we actually have these markers known as MHC class 1 and MHC class 2. And I think of them as a platter. They present. So I might have something that says, you know, this MHC class marker is presenting, well, first of all we need to know MHC class 1 presents self antigens. And they're found on all nucleated cells. So if a cell has a nucleus on it, it's going to have MHC class 1 on top. And it's going to be like, yes, I'm fine. This is proof that I'm fine. So this MHC class 1 is going to present an I'm fine antigen. If this cell is compromised, like a virus gets into the cell and starts making viral proteins, it's going to present something weird. It's going to present either no antigen up top, or it's going to present a weird mutated antigen. And so this cell will present an MHC class 1 marker with a sign saying, I am dying, help me, kill me, or I'm not dying, I've been compromised, I am your enemy now, kill me. So that's what the MHC markers kind of do, or the MHC class 1 does anyway. In terms of MHC class 2, these are found in specific immune cells. And so things like macrophages will engulf a pathogen, eat it up and digest it and kill it, but they will keep the antigen there and present it. And it's like, I found a foreign invader, let me go present to a T helper cell. So they have the foreign invader presented in the MHC class 2, they'll go and tell someone who will start the emergency alarm. So the first line defense is breached, the second line is initiated, which involves a broad response in order to eliminate the pathogen through the use of phagocytic leukocytes and numerous non-cellular molecules. So just know your definitions of these cells. I'm sorry, this diagram is all over the place. Yeah, so know your definitions. They're right here. You copy and paste these from the slide deck into like a flashcard maker or on your own Word doc or something like that. I'm not going to spend too much time on that because hopefully you guys know what most of them are. I'm going to go through phagocytosis very briefly. So I just discussed this process whereby a macrophage or a dendritic cell will engulf a pathogen, such as a bacterium. It puts it into a phagosome. Phagocyte produces phagosome. And what happens is that this phagocyte also contains a lysosome. Lysosome, different from lysozymes and tears. So this lysosome is like a little bubble containing like digestive enzymes. And so this bubble will fuse with the phagosome, they'll join together, and what happens is that it actually digests that pathogen and makes it into like waste. Just expels it as waste. And now it's like safe to just go about the day. So if you encounter a pathogen inside of a cell, inside of your body, this little phagocyte will engulf it, kill it, get rid of it. But what happens is that the phagocytes, such as the dendritic cells or the macrophages, will save the antigen and present it on its MHC class II marker and say, look, this is the problem. I've got proof that someone is invading us and trying to hurt us. And they'll go and sound the big alarm. I'm really annoyed by how like these slides have not worked out properly. I might try and change them. So, the inflammatory response. So when damage occurs to tissues, I'm going to skim through this really quickly. What I found helped me a lot in high school was when I was in the shower or just, I would play with my dog or my cat would scratch me and I'd think, oh wow, inflammation, I'm all red here. And I'd think what's happening, like I'd work through the whole process and I'd just go through it. And just going through it a few times helped me understand and remember it. So if you get a cut or a burn or something, like your cat scratches you, a damaged cell will release cytokine. So damaged cells release cytokines, which are signaling molecules. Maybe a pathogen got in, some bacteria got in. What happens is that the inflammatory response is also triggered, and so macrophages may come to the area and release cytokines as well. So you've got mast cells. Mast cells release histamine. So histamine causes that red, angry inflammation. Because what it does is it causes blood vessels to dilate, so become like thicker, and become more permeable or more leaky, and so more blood flow goes to the area. This blood flow means that more neutrophils and other phagocytes will migrate to the site and phagocytose pathogens and debris. And then finally, antigen-presenting cells will present antigen in the MHC class II marker. Cool. Okay, so our adaptive immune system has a strong targeted memory towards specific antigens. It remembers stuff, and it involves a bunch of different cells. So how can we trigger it? We have that whole process whereby pathogen gets in, phagocyte engulfs, presents MHC class II marker, and migrates to the lymph nodes. Remember, the lymph nodes are like an entire system of vessels which kind of link up to these meeting spaces called lymph nodes, which is where the immune cells hang out. So why does it migrate to the lymph node? Because it knows that the adaptive immune cells are going to be there. I used to think this diagram was really ugly, and I was like, I don't like it. But it's a really good diagram. So if we work through what happens, we've got an APC, such as a macrophage or dendritic cell. And what these are the phagocytes with antigen-presenting ability. So APC goes to the lymph node and it presents that antigen in its MHC class II marker to the T helper cell. T helper cell goes, oh my gosh, we've been infected, this is terrible. And so what it does is it sounds the alarm. It tells the B cell, and the B cell goes, oh my gosh, infected, crazy. And so we actually make many copies of T helper cells. So we clonally proliferate. We've activated the B cell, and we also activate the T cytotoxic killer cells. And what the T cytotoxic killer cells do is that they recognize pathogen, which is in the MHC class, not pathogen, they recognize changes to MHC class I. Remember that all cells with a nucleus have MHC class I. So it's pretty much like all cells, except for the red blood cells, have a nucleus. And so all of these cells, if they've been infected, they will present a weird MHC class I. It'll be either no antigen there or a weird antigen. And so it's kind of a sign or a flag to the T cytotoxic killer cell saying, I've been compromised, you're going to have to kill me, put me down. It also activates the B cell, which can make plasma and memory. You've also got the production of memory T cells. So you've got memory T cells here and active T cytotoxic killer cells, memory B cells, and plasma B cells. And these plasma B cells actually make memory, which is really good because it means, they make antibodies. Memory makes memory. And the antibodies will actually bind to a pathogen and neutralize it. So yeah, plasma and B cells generally defend against extracellular pathogens. T cells generally defend against infected cells, cancers, and transplant tissues. So if you have a cancer, it might present a weird flag or an MHC class 1. And so the T cytotoxic cell will go, that's not good, I should kill you. Or if you're infected with a virus, it'll go, that cell's been infected with a virus, I can see its MHC class 1 is weird, it's a weird flag, let me kill it. This is a really good slide to like revise with, so I recommend going through it. Let me check how many slides we're at. Okay, we're getting better. Let me see how many slides we have. Okay, up to about slide 100. So we've still got like 40 slides left to do. So plasma B cells and antibodies have several roles: agglutination, opsonization, activating complement, which has the similar role as antibody, neutralizing and promoting inflammation. So I had a teacher describe this to me in undergrad as, actually I had it for class the other day. Opsonization is making something tastier. Like bread is wonderful, like I love bread, but if we add like butter to it, it's even better, right? So it's making it tastier. And that's what opsonization is. It's coating a pathogen with antibody and making it tastier for the phagocytes. Agglutination is just a clumpy sort of word, right? A glut sounds like a clump. That's what agglutination is, clumping the pathogen together. So you can see here that the antibodies have two arms. So this one arm clumps to one pathogen, the other arm clumps to another pathogen, which means they're all kind of sequestered. It's just sequestered like 5 times a day, but they kind of like shunt into this big large clump, which makes it, once again, more accessible for the phagocytes, or phagocytes to come and eat. Activating complement. So complement actually produces something called the membrane attack complex, which makes holes in a pathogen. So you can see here, we just put a hole in it, kill it. We can also neutralize the pathogen. So a virus needs to bind, obviously. So if we actually cover all the binding receptors, it can't bind. So we actually prevent that binding. And promote inflammation, which allows more blood flow, makes it more leaky once again. So cytotoxic T cells, I already told you guys, it's kind of recognizing MHC class 1, which has been compromised or changed via these T cell receptors, and it induces death via perforin or granzymes. So we've got the naive T cells activated, it just shoots it dead with perforins and granzymes. I think of it as like when you perforate a piece of paper, you shoot a hole through it. It's kind of what these cytotoxic T killer cells are doing, the T lymphocytes. They shoot a hole through it. And that's what granzymes do also. Okay, monoclonal antibodies are when you... I'm going to have to go through this in a bit more detail. Once again, this will require more detail on your part as well. I just have a really basic guide here. But pretty much, we have pathogens. Pathogens have multiple antigens on them, on their surface. And so antibodies bind to different antigens, which are known as epitopes. So one antigen may have multiple epitopes, so different like kind of similar versions of the antigen, which antibodies bind to, and some bind more tightly. So we can actually, this is called a polyclonal response, but we can actually target the best binding one and create a monoclonal antibody, which specifically targets the epitope, which has a stronger binding affinity. That's what monoclonal antibodies are in summary. That's not enough, okay? You guys can't just go to the exam and go, oh yes, I know what a monoclonal antibody is. You need to know how they're made by combining, I think it's B cells in a mouse with, you combine B cells in a mouse with some kind of viral genome or something like that, because it actually replicates by itself. So if you look up monoclonal antibody, and just go into like images, we use a mouse model. You use cancer cells, that's what it is, not pathogens. So cancer cells pretty much live forever. So we can actually get B cells or plasma cells from a mouse. So here's a good diagram here. Plasma cells from a mouse, and you combine it with cancer cells and you make a hybridoma, okay? That's what it's called, hybridoma. And you make many copies of B cells. So once you've isolated the one which has the best specificity, you inject a mouse with an antigen. So you might give COVID antigen to the mouse. The mouse gets sick. It produces B cells, which make memory. You combine this with the tumor cells, and you've got a monoclonal antibody. So these antibodies won't give you memory, but they will target a particular disease. Okay, so you want to go through these diagrams and try and be able to explain it. Okay, it's a really cool process. Maybe look into a bit more detail on like a YouTube channel or something like that, or an article. But yeah, really interesting process. I just didn't have time to go through it today. Once again, these are very fast, so we've got different methods of identification of a pathogen. PCR we already talked about. Serology is pretty much blood tests. And ELISA is when we actually have like a sample put into a test tube, and we actually combine it with a protein of interest, and that protein of interest will actually bind to a fluorescent protein or a colored protein, and that will bind as well. So you have several washouts. So you add the pathogen. If the pathogen is present, it will stick to the walls, which are primed to accept the pathogen. Then you add like fluorescent protein or something like that, and so you form an antibody antigen complex. And so you wash it. If it's still there, it means it's bound, and so you can like fluoresce it and you can see the color. And if the color is there, then it means that pathogen was identified. I just kind of butchered that explanation. It's definitely a lot more sophisticated than that. So once again, I recommend looking that up. We've just got like 30 minutes left, and we still haven't gone through genetics or exam prep yet. So try and skim through it a bit faster. I don't have a lot of slides on this stuff, I think, on different types of transmission. But knowing that respiratory is like breathing, droplets is like someone sneezes, and you like breathe it in or touch it and eat it. Contact is literally physical contact. Vector is via a vehicle. So for example, malaria is transported via a mosquito. That's a vector. So the mosquito doesn't actually give you the disease itself. It carries the Plasmodium parasite, which gives you the disease. And then sexual transmission, also known as venereal. So these are like STIs and things which can be transmitted sexually. You've got the latent and incubation period. So latent period is where you don't actually see any symptoms. This time is when you start becoming symptomatic. I don't know why it says latent here, and then I'm pretty sure this is when you become symptomatic. Incubation is where the pathogen is like replicating inside of you, and then you've got the actual disease presenting. And so throughout this time, you actually have a period of communicability, which means you're infectious. So even if you don't have symptoms yet, you still are infectious. Okay, this is just on mutations. So we have many different types of mutations, which you guys must know. So point mutations are the substitution of a single DNA or RNA base. See here, this is the wild type DNA, which is null, and then we have what do we have here? We've got the substitution. This is a silent mutation, and what this means is that we actually have a DNA triplet being, one individual nucleotide from the triplet is subbed out, and so as a result of this, you actually have no real change that you can observably see. So because this GTT is kind of the same as GTA, they're both actually encode for valine. So this, like both triplets encode for the same amino acid, valine. So it's known as a silent mutation because you don't actually see any difference in the amino acid chain. In the case of a missense mutation, what you have is a base being substituted or swapped for something else. And as a result, you actually do have a change to the amino acid. So you have a different amino acid being produced, okay, as opposed to what was originally there. So it can be a small mutation or it can be quite a big one. Nonsense are really quite detrimental because what you do is you actually have the production of, you've got substitution. It produces a DNA triplet, which produces a codon. And this codon, if you can see, UAG, you are gone, from our little couple breakup story. This means that we've actually produced or introduced a premature stop codon here. And as a result of this, this entire sequence will truncate a lot earlier than it's supposed to. And so this might actually continue on for quite a long time, but you've actually shortened the protein drastically. And this can be very detrimental. And often mutations like this can result in premature abortion, spontaneous abortion or stillbirth. Quite detrimental. So insertion or deletion mutations are a bit bigger. They usually result in a shift of the reading frame. So if we insert some nucleotides here, it means that all subsequent amino acids are changed because now the triplets are changed and therefore their codons are changed. They can also inadvertently also introduce a premature stop codon. But also just noting that all these amino acids are changed, and so this can actually really affect the function of the protein and be very, very detrimental also. So try and grade these in regard to severity or how detrimental they may be. I just went over the frame shift one. Block mutations, these are where they affect the actual chromosome, like they are mutations on a chromosomal level. So you might have an entire deletion of an entire part of a chromosome, you might have duplication of it, you might have inversion where it's like one bit is flipped over. Here you've got insertion, so this bit of a chromosome is actually inserted, so you've got to swap between chromosome 4 and chromosome 20. And then translocation is where you've actually got an entire segment of one chromosome and an entire segment of another chromosome swapping. Okay? Making sure you understand these once again, and recognizing that these can result in severe like developmental disabilities and syndromes. Okay, how long do we have? Okay, 25. So, natural selection questions. So, I've given you guys like a really good guide as to how we should tackle these questions. So, natural selection questions are very common on the exam. They come up every year. And kind of like a hack I've given you guys is to follow this format I've given you right here. So, firstly, you want to state that variation exists in a population. So, whatever question you get, they're going to talk about, wow, these crabs versus these crabs, or these jellyfish versus these jellyfish, or these frogs versus these frogs. What you want to say is there are two different types of frogs. Variation exists in this population. Some frogs have this trait, some frogs have this trait. Step 1, done. Then we have a selection pressure acting on the phenotypes of the members of the population, so that some members are more likely to survive and reproduce. Something I want to note is for a long time, I was just like, I didn't really understand natural selection when I was in high school, and then I got to bio class and I was like, wait a minute. So natural selection is when you've got a bunch of animals, and some animals have a trait which is seen as like undesirable in a certain environment, and as a result, they die. So for example, giraffes which had short necks, they die, which means that only the long-necked giraffes who accidentally mutated to have long necks, they survive, and therefore they pass their genes on. So natural selection is really about some individuals dying and being unable to pass their genes on. Anyway, in the case of this giraffe situation, the selection pressure is that shorter-necked giraffes can't reach the food they need and they die. Well, the dying isn't selection pressure, but the selection pressure is that you need to reach the high food. Making sure you know that selection pressure acts on the phenotype, which is the physical and physiological characteristics. So often appearance, but often something like they might have really good stomach which digests or something like that. So it can be something you can't see. Anyway, it acts on the phenotype. So selection pressure acts on the phenotype. So what happens is that all those short-necked giraffes die. So they can't reach the high leaves, therefore the short-necked ones die. But the ones who have mutated to have a longer neck, they survive and reproduce. 3. The alleles that correlate with the characteristics that give survival advantage are therefore more likely to be passed on and increase in frequency over time. So whoever had a longer neck, they survive. Therefore, we actually increase the frequency of these alleles over time. So we actually influence the genotype of this population. Over time, the population evolves to suit their environment. I'm not going to go through this, but we can go through that format once again. So variation existed in the resistance of the mosquito. So we talked about mosquito variation. Some mosquitoes are more resistant to pesticides than others, or insecticides. The insecticide is a selection pressure. Anyone with a weak or less resistance, they die. Therefore, those who are more resistant survive and reproduce. The allele for resistance increases in population so that over time, mosquito population becomes more resistant to insecticide, therefore influencing the genotype. Okay? So just use this kind of format for your natural selection questions, and it should help you a lot, I would hope. Selective breeding. I don't like selective breeding. So for instance, pugs have a short snout, which is not good for them, and it can actually interfere with their respiratory systems and make them die early and give them diseases. This is because once upon a time, someone decided that a flat-nosed dog who accidentally mutated looking like that was a really good idea. And so they like bred a bunch of them, and it resulted in these pugs which are really cute, but also it's really bad for them. And so we acted as a selection pressure. We picked a trait we thought was cute and just bred them, which is not good. It also, doing this can actually decrease the genetic variation of the gene pool, and it's like playing God, which people don't like. Selective breeding, I don't really have much here apart from like, we've got different parts of the broccoli flower or the wild mustard plant, and we can take them and grow them and selectively breed them in order to produce different vegetables. How many more pages do we have left? Okay, we can do this. We're nearly done, guys. What is gene flow? It's a movement of alleles between populations of the same species. When gene flow exists between populations, the population's gene pool become more similar over time. Once again, bolded words are things you should know. Genetic drift are changes in frequencies between one generation and the next due to chance events. We've got a bottleneck event or effect, which is whereby you might have a natural disaster which wipes out a bunch of the population. As a result, we might have had orange cats, green cats, and red cats, but we had a natural disaster which killed a bunch of cats. Over time, the new population regrows, but it's like the variation has gone through a bottleneck. We had so many different variations, and now it's smaller. So the surviving population is a lot smaller. That's the bottleneck effect. And then here we've got the founder effect. So if you looked at this is like a big group of penguins. We've got purple and red penguins. But the new population, like they might move to an island. You might have more purple penguins reproducing. And over time, you look at the new island and go, wow, purple penguins must be the only thing that existed back then. And then you go, if you look at the original population, you go, wait a minute, there are red penguins too. What this kind of represents is that the population of a new founding population may be different to the original population of the parent population, and it may not accurately represent the parent population. Like, for instance, I think in Tasmania, there's a high prevalence of Huntington's syndrome disease. And so if you looked at Tasmania, you go, wow, all over Australia, people had Huntington's. It's actually quite rare, I think, and so that population doesn't really accurately represent the parental population of Australia. Okay, so 20-ish minutes left. Bacterial resistance. So, once again, this is a topic I advise you guys to do research on. I think it's insane that this is not regulated. So, I think it might be regulated, but not tightly enough. So, you know how your parents often say stuff like, oh, we must buy like organic chicken because the other one's full of like pesticides and chemicals and it's really bad for you. There's actually truth in that. So, often farmers will feed their agriculture with pesticides and things, or antibiotics, for instance, okay? They feed them antibiotics to help them grow strong and defend them against pathogens and things. But what this does is that it introduces bacterial resistance. So I talked about that mosquito example a few minutes ago, where the mosquitoes become resilient to the pesticides, and as a result, only the more resilient mosquitoes from the pesticides breed and survive and pass the genes on to the next generation, creating like a superpopulation of super strong mosquitoes. The same thing can happen with bacteria. Bacteria has variation. You expose them to a selection pressure of antibiotics. Anyone too weak dies. Anyone strong enough to overcome the antibiotics is left behind. Therefore, they reproduce. Therefore, a new population has strong antibiotic resistance. This is what happens in bacteria. If we eat that, we have strong antibiotic resistance in the bacteria, which means that if we get sick with a disease from the bacteria, we might not have antibiotics to treat that, okay? So it's really detrimental and quite dangerous. So this is why it's a really big deal that farmers and stuff are using, or even, overprescription of antibiotics is occurring. It can actually really affect your bodies and mean that when you do get an infection with bacteria, we might not have the drugs to treat that because of the overuse of antibiotics concurrently. So, something we really want to be aware of. Something else to note is antigenic shift and drift. So, antigenic shift is whereby we've actually got the mixing of a virus with another virus. So, for instance, COVID might mix with the HIV virus, for instance. And so they swap proteins and it creates a new strain of COVID, like the beta strain or the gamma strain or whatever we're up to, I don't remember, which makes it hard for us to create vaccines because vaccines often rely on antigens on the cell surface. And if COVID's constantly using antigenic shift to swap out its antigens, it might mean that we're having difficulty actually identifying the correct antigen to target with our vaccines. Antigenic drift is a long-term accumulation of mutations over time. So you might have one virus and over many, many years it just randomly mutates and then has slightly different antigens. So antigenic shift is a lot faster than antigenic drift. Another question on allopatric speciation. This is where you have, for instance, an island with some capybaras on them, because I really like them. Over time, you might have geographic isolation. So say a river occurs on the island, preventing the capybaras from, segregating the capybaras and having two groups of capybaras. These ones are exposed to sunshine and lots of food and no predators, whereas these ones are exposed to, apparently pelicans really like trying to kill capybaras. Pelicans, which actually happen to eat the capybaras and kill them. And so the capybaras, which, are strong enough, are big enough that the pelican can't eat them will survive here. And so they get bigger, or maybe they can also blend in with the ground. They can't see them as well. So these capybaras on the predator island, those who are like too small, they die. Those who are big, they live, right? They live, and they also, have camouflage patterns, and so they blend in with the ground really well, and they don't get eaten. So they have like all these different changes occurring. Maybe they smell really bad and the pelicans don't like them, or they taste really bitter, just random mutations. So say we're speeding up time, like hundreds of thousands, even millions of years. And so over many, many, many years and many, many, many different generations, these two capybara populations become totally different. This one's been exposed to no predators. They've just flourished as they are. These ones have been exposed to predators and have had to like deal with this. So anyone who's been too weak has just been killed off. But anyone who's had a mutation, which means more growth, more resistance, more toxic to the pelicans, they survive. And this is a weird example, but anyway. If we bring both populations back together, they might be completely different species. And so we know that the species definition entails two populations being brought back. The species definition entails two populations who are unable to reproduce and produce fertile, viable offspring. So they try and reproduce and they can't produce fertile, viable offspring. They are two different species. So over time, we've actually created two different capybara species. Something else to note is donkey and a mule and a horse, I think. Is it donkey and a mule? I can't remember if it's donkey and a cow, donkey and a horse, something like that. I think it's donkey and a horse. You try and breed them together, they produce a mule, which is like, oh my god, new species, or same species, because they can produce fertile, viable offspring. If a donkey and a horse can reproduce and produce a mule, maybe they are the same species. But a mule is not fertile. You can't get another mule and breed them together. Therefore, a donkey and horse are two separate species, because if they're the same species, they could reproduce. Something else to note here is sympatric speciation. So you might have some flower plants growing right next to each other, but they don't breed with each other, or they can't breed with each other because of differences in like temporality, behavioral, or sexual differences. So for instance, you might have this flower flowering in the summer, whereas this one only flowers in the winter. So even though they could hypothetically breed with one another, they're separated by this temporal distance. Okay, fossilization, general traits, like understanding these ones. These come up in the exam a lot. You might need to list like 3 of them. Know them, they're really good. Just no oxygen, alkaline environment, no scavengers, no predators taking the bones or whatever. High pressure sediments and hard body parts. Transitional fossils are pretty much fossils between two species groups. Like when this species becomes a species, this is like a mix. This is the transitional fossil. Relative age is where we kind of estimate age by comparing to other things. So we can use the position or the superposition of it. So if it's between one really old rock layer and one not so old rock layer, we can say it's between there, kind of like middle of that age. Stratigraphy is estimating the relative age of a rock strata by using the layers and some other igneous strata as a reference. And indicator fossils are saying, okay, I know that this creature only existed during this time. If I found this other creature existing at the same level, I'm going to say they both exist at the same time, okay, using an indicator fossil. But we also have more like concrete absolute forms of dating known as absolute dating. This is way too complex to put on my slides here, but it pretty much measures the level of a radioactive isotope within the actual fossil and compares it to the non-radioactive isotope or the stable product. And it's highly accurate, and so it can date stuff. Something you will need to note is that we've only got like 12 minutes left. Something you will need to know is the limits of the dating. So some of them can only be used to date organic stuff within 50,000 years, or some of them are 100,000 years, or some of them 20,000 years. Make sure you know that and know the different isotopes and how they degrade. So carbon-14 degrades to nitrogen-14. That's something you must know. I can't remember the other ones you must know as well, but they are in the study design, so do have a look at those. Oh, okay, potassium-argon dating. So, I don't know if these slides will be a bit messy for you, but I'll put this slide up for a second so you guys can, like, take a screenshot of it, or pause the video and rewatch it here. But potassium-40 to argon-40 is another method. Yeah, once again, these slides are too sparse here. I just don't have enough time to go through all of this, so hopefully you guys make sense of that. Oh my god, we've got so many things to go through still. Okay, Definitions are bolded, you need to know them. Homologous, features in different species that have similar structure but different function. So like we have arms, and arms are similar to the wings of a bat, whereas we walk and swing our arms, bats fly. Different. Analogous, so butterflies and bats both have wings, but they actually work in different ways. So they're actually same function, but different structure. They actually have a different underlying structure. And vestigial structure is a structure that's fully functional in an ancestor, but no function in the current organism. It's like whales have a pelvis because they used to walk on land, which I find a terrifying thought. But they're no longer needed, so it's kind of shrunk a lot smaller. And we have an appendix, which we don't really know what it does, to be honest, but we think it's a vestigial structure for like digestion or something. In order to determine molecular homology, we can use DNA sequencing or amino acid sequencing to determine either DNA sequence or amino acid sequence. This one is more specific because we know that we have silent mutations. Silent mutations would appear as non-apparent in amino acid sequencing, whereas they're quite apparent in DNA sequencing. Phylogenetic trees are pretty much crafting maps which kind of say how related species are and shows where they diverged. So you're expected to know how to draw one and identify, like fill in the blanks in a divergence tree thing by like DNA similarities and differences. My advice for this is just to do the VCA exams and see how they do it, because they kind of do a similar thing every year. Okay, you've got quite a few things you must know. I know I'm going through this fast, but I'm really sorry, guys. Yeah, just put your questions in the chat right now so I can have like 10 minutes to answer them. You must know that mammals have sweat glands or mammary glands as well, fur or hair. Primates have these traits, which mammals also have. Must know that hominoids and hominins are sort of different, or like categories. I'm really not spending too much time on this. You have to spend a lot of time on this. You must be able to detail the differences, which I know can be a bit tedious. Something else I recommend too is also consulting that Q&A doc or your textbook as well. Hominoids and hominins and things are quite confusing, and it's really good to look up hominoids versus hominins, VCE, on Google, because different anthropologists and archaeologists have different definitions. So this is something I'd recommend you guys go and look up, using VCE on the end of it, okay, so that you get what you need for the exam. We have different timelines of evolution. In conclusion, we don't really know which one of these is true or if they are true at all. So many of these relationships are just inferts. We don't know which one is true. So many people think we might have evolved from Homo erectus, but we could have evolved from Africanus or something instead, or habilis before then. So we don't really know. Bipedalism is a trait of walking on two legs, and we've got a bunch of different traits which kind of are conducive toward this. So that arched foot was conducive to bipedalism. We've got these curvatures in our spine, which kind of ensure that our center of gravity is between our 2 feet, which kind of also facilitates bipedalism. We've got this big hole in our skull called the foramen magnum. It used to be very posterior, and it's kind of migrated more forwards, which also confers bipedalism. And then the shape of our bones and pelvic structures. We can also see the changes to skull morphology. So our cranium got bigger, we got less of a pronounced brow ridge, cheekbones decreased in size, teeth got smaller and less prognathic, which is where they kind of stick out. But which hominin species doesn't fit the trend of increasing cranial capacity? We can see that I don't think it's here. Anyway, I forgot the word, Neanderthal has a larger brain than us. And so archaeologists and anthropologists say, oh, it's just because they were bigger. They had bigger body size, therefore bigger brain and bigger cranium. That's a theory. I mean, for all we know, they were smarter than us. But if they were smarter than us, maybe they would have been here instead of us. Anyway, in conclusion, we don't really know why. We think the Neanderthal had a larger cranial capacity than us because it just had a bigger body built than us. Here are some just general traits about each of these species. So Afarensis here, Paranthropus here. That's a sagittal crest there, like a little spike on their head. Know these differences and just general traits. Don't memorize these numbers. Just know that, like, as we progress further toward our current state, like as we go from Africanus to Habilis to Erectus to Agastor, blah, 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 all the way to us, we can see that our cranium, might not look like it, but our cranium gets bigger, our jaw gets less prognathic, our brow ridge gets smaller, our zygomatic arches or cheekbones get less pronounced, our nose becomes bigger, face becomes flatter, and our brain size becomes bigger. Those are our main traits to take away. And then knowing stuff like Erectus was the first, we think, to stand on two legs properly. Or Habilis was like the handyman and started building houses and things. Yeah, so larger brain size than Homo sapiens. We also had these other Homo species which like coexist at the same time. Also, we don't really know, to be honest. There are many theories about them. Homo floresiensis was found in Indonesia, the island of Flores. Small size, we think it's hobbit, we think it might have been humans with some kind of developmental disability, which were all really small. We don't really know. We had a few others as well. Something else to note is that Homo sapiens and Homo neanderthals, we actually share DNA with Homo neanderthals. Human beings as a species, like if you get a human being and another human being and like mix them, they might breed and produce fertile, viable offspring, right? Human beings can produce fertile, viable offspring. But we have Neanderthal DNA in them, which means that maybe a Neanderthal and a Homo sapiens bred and produced fertile, viable offspring, which gave birth to the rest of the human race, right? Because pretty much everyone has Neanderthal DNA. What does this do? Well, it calls into question our theory of a species. We say that a species is two individuals who can reproduce to produce fertile, viable offspring. If they can produce fertile, viable offspring, because we actually have Neanderthal DNA, this means that our definition of species is possibly wrong. Like, this is just a weird grey area in biology that we're a bit confused about. So maybe there'll be a new definition of species, or maybe we'll say that Homo sapiens and Homo neanderthals were the same species. I don't know. In conclusion, we coexisted and we have that DNA, so think on that. 5 minutes left. Okay, mitochondrial DNA only handled from the mother. Therefore, we can actually trace back the maternal DNA all the way through to our initial, like the mother of all mothers, mitochondrial Eve. She was like the mother, the original mother. We have two theories as to how they, like humans, spread out across the world. Either they moved out of Africa and then migrated across the world, or they evolved around the world at the same time. I believe in this one. This is a very vague definition, so definitely go and investigate these further. But yeah. Okay, like 5 minutes to prepare for your SACs. Not your SACs, your exam. SACs are done. I'm hoping you guys can put that out of your mind. Like, I know I was always in the forums like, oh, if I got this mark in a SAC, can I get this grade? Don't think about that. Just do your best on the exam. As I mentioned, I attended these practice exams. Sorry, I'm reading the screen. These revision lectures when I was in high school. And I personally would do really well in my SACs in bio, but I wouldn't do that well in the, like, memorizing them or understanding them. I've just memorized them in the short term before my SAC, and I just forget it all, like, instantly, which is a very bad way to study. So I actually attended one of these ATAR notes lectures, the bio one at RMIT, and I remember sitting next to this kid, and he was like, he knew every answer to every question, and he'd done 30 practice exams already, which he told everyone, and he was like laid back and so chill about the whole thing. And I was freaking out because we did this big Kahoot on the big screen, and I got like 0 questions right. And I was like, wow, I've done really well all year, but I actually don't understand anything, and I'm not going to do well in the exam. And so I was sort of freaking out. Anyway, this kid next to me just kind of spurred me on and was like, this kid is what I should be like. I need to do better than this kid. And so what I did is I pretty much studied the entire study design, the entire bio study design from like front to back. These are all just general tips. And you can't see that one. I'll just show you what it is. Oops. There you go. Yeah, so I just got the entire study design. Here are my tips. And I just like printed out all of it. So I printed out the whole thing and laminated it and put it in my room. And I went through every single dot point in the study design, and I bought these little summary books. They were the A+ bio notes ones, if you guys want to see what they look like. I don't think you can get them on the ATAR notes website, but the ATAR notes book is also really good. So I bought this bio book, and I annotated it, and added my mnemonics, and drew my little breakup couple mnemonics and stuff, and just added all of these things to it, so I could, like, remember stuff. So I read this entire summary book, and I went through my textbook, and the textbook I loved reading, I had the Heinemann one, but it just had too much info, so I'd read what I needed, and I would just relearn every single point, and I would make a point of teaching my siblings, or my parents, or the cat, or the dog, or the stuffed dinosaur on my drawer, or whatever, and just try and teach it to someone, and explain it, because in essence, what you need to do is explain to the examiners, okay? So making sure that you can explain it quite confidently is really important. Annotate your notes. I'm a really visual learner, so I need drawings of a couple yelling at each other, or I don't know, a brain or like a fossil or whatever. Like, I need these pictures. And I need to draw these pictures, and I need to make mnemonics, which are weird with strange storylines, because that's how I learn, and that's how I remember stuff. If you learn stuff from listening to a podcast, go out and listen to the VCE Bio podcast, or, watch YouTube videos and listen to them on the way to school. Or if you learn by flashcards only, make flashcards. Figure out how you learn. I was always so, gaslit into, watching videos in high school. Everyone was like, oh, watch videos is the best way to learn. So I would watch videos, and my brain often just checks out after a while, and I don't absorb anything, which you guys might be doing right now. So take a break, come back. But I found I can read a textbook and understand stuff a lot better there, because I've got to stop, read, make sense of it, draw a picture, diagram, whatever, and then get back to it. So figure out how you study and do that. And then find as many practice exams as you can. So I did 20 bio exams, and I ended up getting a 49 study score. I personally think 15 plus is a really good exam number to aspire for. I think if you get over 15, you should feel pretty confident about the exam. So do as many as you feel is suitable. If you do 20, I feel like you feel really well prepared. You've counted so many questions. Vika obviously has a list of 20 years worth of content. You have different company exams, NEEP, ATAR, NERS, QUATS, whatever. Your teacher might have a bunch to do, so seek them out, ask them for exams. If you have a tutor, ask them for exams, and do these exams. What I did as I was doing the exams, I printed them all out, covered my entire coffee table, and every time I got a question I was unsure about, I would circle it. Every time I got a question wrong when I was marking it, because I would mark it myself, I would also circle it. Every question I circled, I would put it into my notebook and write a list of why I was confused, what the real answer was, why I got it wrong, or even if I didn't get it wrong, why I was confused about it, and make sure I fully understood it. And I would read this little errors book every night before bed, and just consistently okay, let me remember that, shorthand repeats, blah, 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 how they work. Stuff like that. So make a note of every one of those questions, go back and study it, revise it, put it on the ATAR Notes forum. This is when I started using the ATAR Notes forum. So I would every time I got a question wrong or was unsure about it, I would take a photo of it, put it in the forum, be like, this was my answer, I don't know why I got this, or what the real answer is, help me please. And some person would help me out, and it was really nice. So if you just use the ATAR Notes forum for anything, do that. Do more exams, revise your notebook, do more exams, review every day. And I actually walked into the bio exam. Maybe this just shows what a nerd I was, but I was looking forward to it because I'd done so many practices and I felt so confident in my ability, which sounds like such a nerdy thing to say. But you've got to be aware that I just relearned the entire bio syllabus in the September holidays because I'd spent all year just wasting my time memorizing it the short term. So I pretty much had to I couldn't even tell you what transcription was. So I had to relearn that and translation and everything else. So yeah, you guys can start now and still kill the exam, in a few weeks, which is what I did. So yeah, good luck, you guys. Things to take away from this lecture: make an ATAR Notes account, do practice exams, 15 to 20 of them, put every question you got wrong into the account and on the forums and someone will reply in the bio discussion forum. What else was my key takeaway? I have some sample questions here, those natural selection ones, allopatric speciation. All of those are good to use as a reference. Yeah, and then I did give you a list of things to go back and study further because I did have to cover them very briefly, which is a bit sad, but yeah. Hopefully you guys took something away from this lecture. Thank you so much for coming. I really enjoy these lectures and I'm glad you guys come to them, so let me know if I helped you out in the comments or the discussion or anything. And yeah, good work guys. Lots of study and you're making the most of your holidays, so just a small grind and you'll be there. So good work guys, have a good holiday and good luck for your exams.